Good afternoon and welcome to Denver for the 2024 annual meeting of the American Society of Human Genetics. This year's meeting promises to be fantastic, filled with cutting-edge research in our field. Having lived through the years of the COVID pandemic, it still feels like a true privilege to be able to gather together in person. One of the special powers that comes with having the honor to serve as the ASHG President is the ability to know what the attendees of this year's meeting are thinking. I realize that the overlap with the US presidential election will be a distraction for many of us, possibly for the entirety of our meeting. I hope you will join me in trying our best to not let that diminish our engagement with the many scientific opportunities here for us this week. Before starting with my scientific remarks today, I wanted to accomplish two things: highlight the achievements of the Society so far this year, and to thank the large number of persons who have made those advances possible. In 2024, we identified our new CEO, Amanda Pearl, who started in May. She has quickly learned about the ASHG and is moving us forward, implementing our new strategic plan completed last year under President Brendan Lee. If you see Amanda over the next few days, please introduce yourself. With increased support from the NHGRI, the ASHG was able to increase the size of our fellowship program with the addition of the ASHG, NHGRI, Genomic and Public Service Fellowships, and extend the funding of the Human Genetics Scholars Initiative through 2027. Shown here are some of the members of the class of 2024 for those programs. Given the increasing threats to NIH funding and the uptick in anti-science rhetoric, the ASHG has increased its advocacy activities, focused through GPAC, chaired by Tony Winshaw-Boris. Dr. Chasman Jackson led the ASHG in convening the first summit with partners from historically Black colleges and universities, hoping to facilitate human genetics and genomic research on their campuses and to improve diversity in our human genetics research workforce. Stay tuned for more to come. In the publishing arena, the Society successfully extended the AJHG's contract with Cell Press. We also initiated a search for the next AJHG editor-in-chief, as Bruce Korf will finish up in about a year. We hope to be able to announce the next editor in a few months. This year, we are celebrating the 75th anniversary of the AJHG. Look for related events here at the meeting. Among the many, many individuals to thank for their devoted efforts for our society, I want to particularly note the program committee chaired by Beth Sullivan, who worked so hard to pull this meeting together for us. My gratitude to the ASHG staff, now led by Amanda, who enable all that we enjoy from our society year-round. My great appreciation to all of the members of our board of directors and our executive committee. Their enthusiasm and thoughtfulness were again apparent at our board meeting yesterday and today. A big shout out to the many members of the various ASHG committees and working groups. They are the ones who enact our many strategic efforts. My huge thanks to our two editors-in-chief, Mike Bamshad and Bruce Korf. Both have advanced the agenda for the ASHG's two journals under quite challenging and uncertain times. We have been so fortunate to have the NHGRI under the leadership of Eric Green as a trusted partner. We've expanded that partnership this year and believe even more is possible in the future. Finally, my deep gratitude to all of you, the membership of the ASHG. Over the years, but especially this year, I've experienced the passion that you bring to our field and to our society. We are so fortunate to have one another, whether to collaborate scientifically, to disagree with each other as required for scientific progress, or to band together to fight for the proper use of human genetics and genomics in society. Thank you so much for inspiring me. If one reviews the types of addresses that past ASHG presidents have delivered, there is a fascinating range. Adopting the approach of the first ASHG president, Herman Muller, which has since been used by many of my predecessors, I decided to speak about a specific topic in human genetics. As per my talk title, I will be discussing the related concepts of incomplete penetrance and variable expressivity. In the next several minutes, I hope to explain why I believe that these old genetic ideas have taken on new urgency and provide a vision of the important opportunities in human genetics and precision medicine deriving from current challenges. This slide may only be needed for my wife, who is here, so bear with me. I promise that she would be able to understand this talk. For Mendelian disorders, so-called single-gene traits, penetrance is the probability that an individual with a pathogenic variant develops the related disease. If it is 100%, we label that as complete penetrance. If it is less than 100%, penetrance is deemed incomplete. Expressivity is defined as the degree to which trait expression differs amongst affected individuals. If it differs, we call that variable expressivity. And of course, traits can exhibit both incomplete penetrance and variable expressivity. Please note that I took the graphic display of these concepts from Carolyn Wright, who's an insightful thinker about them. Her name will appear more than once as I move along. As you will hear in just a few minutes, the concepts of penetrance and expressivity are about 100 years old. So what factors are driving the urgency now? As shown here, I believe that the combination of the falling price of genome sequencing, an amazing and enabling force in human genetics, and the sometimes misapplication of penetrance and expressivity is putting us on a collision course. As we seek to leverage the former, the latter is creating challenges for the robust enactment of precision medicine. Let me illustrate the potential problem with a thought experiment. An experienced group intends to advance precision medicine through genetic screening of the population. They assemble a list of genes to be screened and the relevant information as shown here. Of course, this group has important decisions to make about age of onset and actionability for their list. However, they are likely to insist that the genes on the screening list have relatively high penetrance. This in turn raises the question, how do we know what the penetrance is for a given gene trait pair? The usual answer has been from careful review of the literature about that gene trait pair. In nearly all instances, estimates of penetrance for gene trait pairs are derived as shown here. A cohort with the trait is assembled, probands are genotyped, cascade testing is performed to find additional genotype-positive relatives who are then phenotyped. This allows penetrance to be calculated. I want to propose that we call such estimates penetrance familial. But of course, what the precision medicine team will do is not analogous to that. Rather, the analog is to genotype-first studies using biobanks, for which investigators genotype participants, identify those carrying pathogenic alleles, and then extract phenotypes from the available records. A quick pause to acknowledge that the ways in which individuals in biobanks are ascertained, the criteria used to curate alleles as pathogenic, and the limitations of available phenotypic information are very important factors. For this talk, permit me to assume that all of that has been done perfectly. The resulting estimate of penetrance I will call penetrance population. The crucial question for precision medicine is, are penetrance familial and penetrance population the same? A related issue pertains to the important improvement in human genetic research, normalizing return of secondary findings to participants electing to receive them. This, of course, has relied on the efforts of an ACMG working group, which has been evolving their list of gene trait pairs since 2013. They anticipated the likelihood that penetrans population, which is the relevant one, would be less than penetrans familial. However, penetrans was not included in their methodology for version 1. In version 2, penetrans was added to the methodology, but the list relies on penetrans familial. As you can read from this quote, they understood that this was potentially problematic. This past summer, the group issued a position paper about penetrance, alerting the human genetics community to this issue and thoughtfully discussing the equities. Several Mendelian traits have been studied using genotype-first methodology, attempting to address the previously posed question, are penetrans familial and penetrans population the same? The answer for the traits studied to date is that, in most cases, penetrans population is significantly less than penetrans familial. And if the study is performed using a cohort skewed toward healthy persons, such as the UK Biobank, penetrance is even lower. Shown here is one example, maturity onset diabetes in the young, from a study by groups at Geisinger and the University of Exeter in the UK. As you can see, for probands and genotype-positive family members, penetrance of diabetes amongst HNF4A pathogenic allele carriers is 90% or more. In contrast, estimates for penetrance population are only 20 to 30%. Shown here in the red squares is penetrance of hyperglycemia amongst carriers of GCK pathogenic variants. For this gene trait pair, penetrans familial and penetrans population are equal. This appears to be the exception, not the rule. I have been discussing the importance of ascertainment vis-a-vis penetrance, but comparable differences are found for expressivity. Shown here from a study that Brittany Wenger in my group undertook, she examined heights amongst carriers of pathogenic alleles for Noonan syndrome, a trait for which short stature is a feature. Shown on the left are the results from the healthcare-based biobank at Mount Sinai called BioMe, and on the right are the results for UK Biobank participants. The red line indicates the third percentile for the typical population. Only 2 of 21 carriers, or just under 10%, were below the third percentile as adults. In contrast, 40% of individuals diagnosed with Noonan syndrome have final adult heights below the third percentile. To understand how we arrived at our current definitions and understanding of penetrance and expressivity, I decided to trace their intellectual history. This turned out to be quite challenging. I didn't know, and neither did anyone else whom I approached. Former ASHG President Aravinda Chakravarty helpfully pointed me to a paper by Edgar Altenburg and ASHG First President Muller about a Drosophila wing trait called truncate. Their paper, published in 1920, was a tour de force. As indicated from this quote, they realized that the trait truncate was not Mendelizing. Remarkably, Altenburg and Muller used linkage analysis to map the gene for truncate to chromosome 2, but also showed that there was at least one genetic modifier on chromosomes 1 and 3. Reading this, one realizes that such genetic complexity must have been present with some frequency in the Columbia fly room, but that the group selectively studied Mendelian traits that behave simply. There were, after all, plenty of those, and the complex ones were just too challenging. The issue of simplicity versus complexity in thinking about Mendelian traits will be covered at the Presidential Symposium tomorrow by the historian Greg Raddick. With respect to the history of penetrance and expressivity, this paper turned out to be a dead end. Altenburg and Muller never discussed the concepts per se, and there wasn't any follow-up from them on truncate. A few years later, the interaction of these two men led to the coining of the term penetrance and expressivity. Many of you will recognize the person on the left, Vladimir Lenin, the founder of the Soviet Union. Probably few will know the man on the right, Oskar Vogt. Vogt was a German scientist, specifically a neuroscientist, as you might infer from what he is holding. He pursued a structure-function paradigm, believing that the minutiae of brain anatomy would explain various neurologic disorders. Vogt was one of the best neuroanatomists in the world in the 1920s. During this interwar period, the Germans and Russians were collaborating extensively scientifically. So, in 1924, when Lenin died, the Soviet authorities summoned Vogt to Moscow to dissect Lenin's brain with the question, why was Lenin so brilliant? If that sounds laughable, recall that something similar was done with Einstein's brain 30 years later. When Vogt arrived in Moscow in 1925 to start his study of Lenin's brain, he was introduced to leading young scientists, notably those pursuing Drosophila genetics, a Russian strength in the pre-Lysenko era. On the left is shown the Drosophila wing vein trait called radius incompletus that a husband and wife team, the Timofey-Frasovskys, were studying. This trait was notable for its incomplete penetrance. The Timofey-Frasovskys outcrossed the trait onto different fly strains, showing that penetrance levels varied amongst the derived lines. On the right is the work of Romashov. He was studying a trait of variable coloration patterns of the fly abdomen called abdomen abnormalis. He showed that the degree of expressivity depended upon the wetness of the fly food, but was heritable. When Vogt got back to Berlin, he did two things. He recruited the Timofey-Frasovskys to his institute, which had dire consequences for them after World War II. Vogt also got both groups to publish papers in the German language Journal for Psychology and Neurology in 1925, and in 1926 wrote a piece in which he coined the terms penetrance and expressivity. Importantly, Vogt made mistakes as he set forth the concepts of penetrance and expressivity. He conflated gene and allele, but more critically, he seems to have missed the fact that the penetrance of radius incompletus was dependent on genetic background and varied from one genetic background to another. Put another way, penetrance was not a fixed parameter. The concepts of penetrance and expressivity were picked up in England through Waddington about a decade later. Interestingly, the concepts did not make it into American genetics textbooks until the mid-1950s, and that was driven by Dobzhansky, who had emigrated from the Soviet Union, where he had known the Timofey-Frasovskys and Romashov. The errors committed by Vogt with respect to penetrance and expressivity have echoed down to us today. To demonstrate that, let us look at the Wikipedia entry for penetrance. Here is that definition. I have underlined the word "the" for emphasis. The implication of this definition is that penetrance is a fixed parameter. And here's the second error descended from Vogt. As you can read from this quote, gene and allele are being conflated, implying that penetrance is equal across all alleles. I will show you in a moment that this is incorrect. If penetrance and expressivity are variable, what are the factors driving that variability? The current best understanding divides the driving factors into three broad buckets: genetic factors, environmental exposures, including epigenetic factors, and stochasticity, also known as randomness. The relative contributions of those buckets to the variability in penetrance and expressivity are generally not clear for human genetic traits. I will briefly discuss these two buckets. Of note, ASHG member Athena Starlin Davenport will discuss genetic modifiers for sickle cell disease at the Presidential Symposium. Stochasticity, which I will not discuss, will be the topic of Stas Schwartzman's Presidential Symposium talk. Shown here is a result from another study from Carolyn Wright's group, looking at fluid intelligence as measured in UK Biobank participants. The dashed line shows fluid intelligence in participants found to harbor no pathogenic variant for a Mendelian developmental disorder. The solid line shows those who harbored one DD variant. As you would expect, the performance for the former group was better than the latter. Then, a polygenic risk score for educational attainment has been applied, with the percentiles arrayed along the x-axis. The impact of that PRS is quite similar for both groups, meaning that the expressivity of those harboring a DD variant depended on genetic interactions with the complex trait represented by this PRS. In fact, the impact of the DD variant on fluid intelligence is equivalent to 20 percentile points on that PRS. Put another way, knowledge of only the allele status for the Mendelian DD trait is insufficient to accurately predict outcomes which actually depend on the more holistic sense of genotype. For common and less common genetic variants affecting penetrance and expressivity, we have deep insights into plausible biological mechanisms through which they might interact with the large effect Mendelian alleles. Needless to say, working out the specifics is far from trivial. Here, I am showing a portion of a figure from a UK Biobank study of penetrance of the Mendelian trait familial hypertrophic cardiomyopathy from James Ware's group in London. These are the estimates with 95% confidence intervals for pathogenic and likely pathogenic alleles of the two commonest genes causing this cardiac trait. As you can appreciate, the penetrance estimates vary widely, from one for which the confidence interval includes complete penetrance to one with penetrance of only 10%. Not shown is one pathogenic splice site variant in MYBP3 with a penetrance of 1%. The need to understand penetrance at the allele, not gene level, is apparent if the promise of precision medicine is to be fulfilled. Now I turn to a brief consideration of the impact of environmental exposures. While we have rare spectacular examples, neurodevelopmental outcomes in individuals with phenylketonuria, depending on diet, being probably the best known one, gene by environment studies for Mendelian traits have been difficult to power adequately, and thus few such interactions are well established. Here, I'm showing a result from Gary Cutting's group, looking at the impact of climate temperature on lung function among individuals with cystic fibrosis. 3 independent cohorts were studied, all yielding similar estimates of the negative impact of increased temperature. How many environmental factors are affecting penetrance and expressivity and their aggregate effect sizes on Mendelian traits remains obscure. Before summarizing, I want to recount another piece of genetic history to make a point about the interactiveness of genetic environmental factors with respect to penetrance and expressivity. As recounted in the ASHG publication, Facing Our History, there was a shameful period in the 1970s when incorrect assertions about the heritability of intelligence and scientifically nonsensical but socially harmful claims of genetic inferiority amongst Black people were promulgated by Jensen, Shockley, and others. While the ASHG regrettably refrained from rebutting those genetic falsehoods, some scientists did step forward. One of them, the renowned evolutionary geneticist Richard Lewontin, made seminal points about how we need to think about the interaction between genetic factors and environmental ones with respect to heritability. He pointed out that the interactions between genotypes and environment can take rather different and complex shapes, forming what Volterek first termed in the early 1900s as reaction norm, based on observations of the crustacean Daphnia. Today, these are called norms of reaction. Lewontin knew well about norms of reaction because his PhD advisor at Columbia in the mid-1950s was Dobzhansky, who was writing about them then. Interestingly, norms of reaction continued to be studied by ecologists, but been basically lost to human geneticists. Here, I am showing 3 potential norms of reactions from Lewontin's papers. Focusing on the one on the left, you can appreciate that the impact of 2 different genotypes, labeled G1 and G2, on phenotype, represented on the y-axis, flips depending on the state of the environment, represented on the x-axis. If environments are randomly distributed around E', then the net impact of environment is 0. However, if either genotype predominates, there is a strong average environmental effect. In the middle figure, genotype barely affects phenotype at the environment at the extreme right, but impacts it dramatically from environmental states on the left. Finally, there's the special norm of reaction shown on the right. Here, both genotype and environment matter across the spectrum consistently. Lewontin's point was that only when this norm of reaction is present can one accurately use the analysis of variance, as per Fisher, that leads to our usual formulation of heritability and environmental variance contributing to complex traits. I recount this because norms of reaction are equally applicable as we come to grips with the complexity underlying penetrance and expressivity for Mendelian traits. Depending on the relevant norm of reaction for a given trait, the penetrance of its disease alleles need not be stable in different environments. To summarize, I hope that I've convinced you that the promise of precision medicine requires that we correct errors in our understanding of penetrance and expressivity made nearly 100 years ago that continue to reverberate among us today. Penetrance varies depending on how carriers of Mendelian disease variants are ascertained, such that penetrance population generally does not equate with penetrance familial. Moreover, genotype needs to be thought of in a holistic sense, not focused entirely on the causal Mendelian variant. Penetrance and expressivity will often be allele-specific, not stable across the entire gene trait pair. Penetrance and expressivity can also be dependent on environmental factors. Norms of reaction can be relevant in understanding how genetic and environmental factors interact, rendering penetrance and expressivity as potentially variable in a context-specific manner. As scientists in the field of human genetics and genomics, we have both challenges and opportunities to enable the full potential of precision medicine. We need to elaborate penetrance population from Mendelian traits for which we feel population screening or return of results are worthwhile. As reflected at sessions during ASHG meetings, we have enormous scientific expertise in methods for studying Mendelian disorders and complex traits, but they tend to be siloed. We need to marry these approaches. We need to parse the relative contributions of genetic and environmental factors to penetrance and expressivity. I am arguing that we need to bring back the concept of norms of reaction to our field and grapple with their importance for understanding Mendelian traits. Figuring out how to accomplish much of this in a robust way in the face of the rarity of Mendelian traits is a tremendous challenge. It will require ingenuity, collaboration, and perseverance. Before closing, I need to acknowledge the persons listed here, all of whom provided concepts that I have presented to you today. Largely, I relied on their ideas, just repackaging them for this address. I thank you again for the honor of serving as the ASHG President this year, and am so grateful for your attention this past half hour. Enjoy the meeting.